1D part, which are wires, 2D, which are surfaces, and then 3D materials, which are like three-dimensional matrices that contain nanomaterials inside it.
At what point in that sentence did you realize he doesn't know what the fuck I'm talking about?
I thought this kind of, the notion is I worked with actually fruit flies. And I was going to use a fruit fly.
Which I have a question about fruit flies. I hate to get
off. That's all right.
Where are they? I know, but let's say like they're not in my house anywhere, right? And then I have bananas, and those bananas go bad, and they start to go bad, and now I have fruit flies
everywhere.
Did they come out of the banana?
Yeah, probably. They probably got laid on. They lay eggs. I mean, if you see a rotten banana, you're eating larvae. If you eat a brown banana, really brown, if you put that banana in the freezer, more often than not, you'll see frozen larvae crawling.
Right, but I mean, that's what I mean. Are we eating the larva the entire time because it's already in the banana?
I think they're probably laying eggs on them.
But where? But they're not flying around my house in general.
Well, they could be at the grocery store. I mean, they're going to lay eggs on anything.
Okay.
And it takes, depending on temperature. If it's room temperature 25 degrees, it's going to take a couple weeks.
Okay.
20 degrees, 3 weeks.
So they come with it on
lower
it.
than that, they could be laying eggs on it. So if there's something right,
Okay, because I was like, these things are just materializing out
it's kind of like
of nowhere. Like, do they time travel?
Spontaneous.
Are they like squirrels where they're like in front of a car and then boom, now they're in my bananas?
But this is an argument people have for spontaneous life, right? They would, this the whole thing back in the medieval ages or whenever they did this, they saw like maggots coming from meat, so they thought they were spawning. Immediately from it. So there's the same sort of deal. And then they put like, cheesecloth over it, and then they didn't get the maggots, just got rotten meat. And then they realized that the eggs were being laid on there, and then they started finding these little corpuscles that were. So I think the same thing with the fruit flies. They're everywhere. I mean, a glass of wine, and they're like vinegar flies. I think this expression, you get more flies with honey than vinegar. Not for fruit flies. fruit flies, they actually like the fermentation products.
You're like, you know what, as a scientist, that joke isn't technically correct.
Yeah, exactly. I heard someone mention it before. I'm like, that's not right. But I didn't want to break up the thing. I think it was my yoga class.
That's great that you think
I'm like, oh God.
That's
wrong,
how my mind.
actually.
I don't even know what. I don't really understand how small a nano. The nano's not smaller than an atom, or is it smaller than that?
No. So angstroms, which are the unit lower, which is like, so nanometers 10 to the 9 meters. So the way I explain to is your hair is like 10 microns. Okay. So the width of a spider thread is like a micron or 10 microns. 1,000 times smaller than that, you have nanometers.
Okay.
So you're looking at, and I think like a nanoparticle of gold has like 400 or 146 atoms. Like it's like a 2 or 3 nanometer particle of gold has a limited number of atoms. So you're looking just above the atomic scale, but just below kind of what we call bulk or where the properties kind of, the nanoscale properties disappear. So if you're below 100, and that's the defines on nanoscience or nanomaterials, any material which has a dimension of less than 100 nanometers on any one side. So you have 0D nanomaterials, which are particles. You have 1D which are wires, 2D, which are surfaces, and then 3D materials, which are like three-dimensional matrices that contain nanomaterials inside it.
At what point in that sentence did you realize he doesn't know what the fuck I'm talking about?
You look like you were a guy. Actually, I see the same expression my students. I'm like, my God, how crappy at up here.
No.
All right, class over.
No. It's just like this level of
test next Tuesday.
That's why this show's called Dumbest Guy in the Room, because that's like I have vague knowledge of things,
well,
and then I'm like, and I'm just like, is most of the research driven towards like business and technology? So like developing it for like products and stuff, or is it more like, hey, we have to save the planet, we have to save the
so there's a bit cancer. So my research is much more fundamental, and I have some stuff which is we're patenting or working with people that patent. Other people are much more applied, and they have stuff they're developing things that are much more commercially available. There's a lot of people starting companies. We have one of my peers, Dr. Ratnakia, she's started a company called Minerva Lithium, which uses nano materials to basically pull lithium out of water. And this is really huge, right? Because then you can recycle it and kind of, and for the big push for electric cars, it's a great way to it's a very powerful method now to kind of capture lithium.
So if you're doing it that way, I'm sure it expands the territory of where you can find lithium too, right?
Yeah, exactly.
Is it basically in all the water?
I think so. I mean, it's an amazing material that she's developed. And that's kind of where this nano comes from, is that you're able to take old materials or develop new materials which have these capacities that, because you've increased surface area, because you've kind of isolated individual ions or atoms rather that are much more actively involved, you're able to kind of get more out of what you have rather than just bulk materials.
This is something that you would be familiar with. I know we're not far off from having nanotechnology, little nanobots in our bodies that will fight disease, that'll give us medication and fight cancer cells and stuff like that. Have you done any work in that?
I haven't done anything. Like I said, there's a colleague at the Nano School. We just hired her. She works on kind of these smart materials, these robots, which can basically target or swarm. And that's one of the big issues when it comes to, the challenges for any type of these treatments. I mean, for gene therapy, for instance, we've had these great ideas for 30 years of how to fix genetic disorders, but the real issue is how do you target cells? How do you get efficient expression of the thing you target? And so these nanobots, so to speak, would enable you to kind of be able to program them to seek a specific cell type, a specific area, and therefore you'd be able to kind of like eliminate off-target, which is a big problem with even chemotherapies, right? The reason chemotherapies, they work, they attack everything, right? Yeah. They're not specific. So, and that's where a lot of nano is going anyway. They're trying to make nanoparticles target. You encapsulate it, you make a nanomaterial that has a slower diffusion rate, but it still contains a high dose of the drug, and it's not going to be mobile. It's not going to be transported everywhere. So there's a lot of, again, it all comes back to why people look at nano, right? Because nano has it's weird little scale, right? It's below 100 nanometers. And that size does a lot of interesting things to materials.
What do you mean?
So like gold. So gold is, this is a classic example. So gold is yellow, right? But if you make nano gold, it's red. And actually, if you look at stained glass throughout Europe or here, a lot of that red color is from nano gold. So they've known it for centuries. That nano, when you get it that small, the way it interacts with light is totally different than the way the bulk material has. And that's true for a lot of other types of interactions as well.
Huh.
And a lot of nanos going, like we talked about the semiconductor. So that's a big push. And one of the projects we have down at the Nano School is using DNA as a storage device for the future. It's like more of an archival storage because it's really slow, but using it as a read-write system where we can store large chunks of data.
DNA is really slow?
Being able to read or write it is. So the technique, it takes, for our cells to express a gene, it's anywhere from 15 minutes, the most rapid, to several hours. Wow. And if you're a cell phone or computer, damn it.
So you were going to use it as like a external hard drive to hold a bunch of stuff?
Tons of, you can just dump data on it. Huh. And it has a high storage capacity because it's a small molecule. It's 2 nanometers wide, right? So you're able to kind of put a lot into a very long, skinny molecule. And our cells put
incredible,
amazing amounts of data into it, right?
I was just reading that if you took our DNA and stretched it out, it would go to Pluto and back a couple times.
It seems like all of reality is just data. Maybe.
Yeah.
It's very likely that it's all just information, and we just interact with this information, and our perception is not really what it is. It's just the way we interact with it.
So it is like that.
Yeah, have you read any of Donald Hoffman's stuff?
No, I haven't.
He's got some. He's working on some theories about that. His team, and he's a reputable scientist. And
no, you can see it. If you think about everything we have, it's just layers and layers of information.
Yeah, Ray Kurzweil had a theory or a hypothesis that everything is really just already nanotechnology. We just haven't realized it yet. It's just self-replicating nanotechnology.
Yeah, in some ways. Because our cells, I tell my students, cells are nano, right?
Right, yeah.
Cells interact at the nanoscale. Everything about the cell is nano. When you're talking about mapping the brain, that's a big thing. The cells in the brain, while they're kind of micron scale, their interactions, how they touch each other, interact with each other to build these bridges, that's all kind of nanoscale interactions that really increase the amount of connectivity between different cells and different neurons in the brain. It creates this amazingly complex organism that we are right now.
Do you every couple of years sort of like get re-excited and be like, oh, this is where we should be going, and this is the angle I want to do, or that thing I thought was going to work is it's like blockchain. I can't really. I know it's great, but I can't figure out how to really use it for my advantage.
I think you would be very nimble in this field because there's a lot of areas where the fundability, even something that's really cool, if people don't quite get it, you're not gonna be able to get the funding for it. But at the same time, you have to, hold towards a certain tack, because you want it to be, if you have a good idea, you should just push for it and fight for it. But there's oftentimes you have to find the direction you go towards. It's not usually a straight path. You have to find other ways.
I feel like we're on a roller coaster, and we're like clicking up the hill.
It's probably
and as soon as we hit the singularity, we're gone. And I think at that point we just hang on, because I think that all the AI and all the stuff that we've already created just starts to just churn out unbelievable amounts of technology. We're probably like, what do you call it? What do you call the technology? It's it changes everything. It's
transformative. No.
Yeah, like transformative technologies. Yeah. But you imagine having those every hour.
Yeah. Well, I think you're right. The AI is a big thing. So a lot of I'm pushing my students to more
disruptive technology.
Disruptive, yeah.
Yeah.
I think that's really where it's going to go. I think you're going to see more AI being involved in the process. And I think the need for it is because when you start looking at these finer and finer things like nano and other areas kind of allied to it, you need to be more consistent experimentally when you start testing these theories and testing these ideas. And you have to have some sort of way of being able to have other labs, because there's multiple people doing this, being able to share information. So a lot of these meta-analysis where you're taking information, you didn't know how people did stuff. And sometimes, and this plagued Nano early on, that people would make these amazing materials, right? And no one could replicate them. And the rationale is because there was something else going on. There was they were able to do it consistently, or at least you'd hope, in their lab. But when it came out, they weren't able to reproduce some of these things.
Yeah.
And
why?
Because it was
so complex.
Or they had something weird, like their lab or their experimental system was set at a different temperature. Or there was some variable control in there which you didn't know. We've had stuff like that happen. We've made some materials like, wow, that's so cool. And repeating it, you find out that there's one time we made these really amazing composite materials out of bacteria cellulose. So like kombucha, right?
Oh yeah.
Like SCOBY is a bacterium and a yeast. The bacterium makes this network of nanocellulose, which is 20 nanometer fibers of cellulose, which is a lot stronger than cotton. And it's really kind of amazing the way it interacts with water. So we found a way to make these composites where you can do, add some salts, some transition metal salts to it, copper, gadolinium. And when you go through a simple kind of thermochemical microwave synthesis process, you can embed these particles in the matrix of those fibers, which opens up a whole new possibility of new materials.
Like really strong, flexible fibers.
Yeah, exactly. Or conductive fibers. So this super cool, or semiconductor fibers. We have stuff made out of this semiconductor material. So it's super cool. But but when my student did this, Ross did this, we needed some ammonia. He wanted to do it fast. So he went to the dollar store, bought some ammonia, and made this stuff, which is like, wow, he was able to grow like copper wires from it. There's other things he was doing. It was super cool. And then we bought ammonia, 100%, laboratory grade. And it worked, but not the same. I was like, what the hell?
So you could fund the school with all that copper wire and steal copper wire and
he's built. The pictures were pretty. I should have. They looked like flowers coming out of this matrix of
stuff. How much was he able to produce from? Like, what was the size of the experiment?
So he was growing petri dish size.
Okay.
Fibers, and then the whole thing would turn brown, and then you could actually that was just making copper oxide, but he could make batches. That was the nice thing about it, is you can make.
So it just rusted.
That's amazing.
Pretty much, you just rusted. And then, but what he did with this was he used, I think, gadolinium, made the gadolinium oxyhydroxide. No, it was a gallium oxyhydroxide material. He exposed it to UV because it's a semiconductor material that sheds electrons. So he used that. And the idea was to kind of have it reduce copper oxide to copper metal. And what he saw were these blooms of like these wires coming out, which is
crazy.
Kind of hard. It was totally crazy. Like, wow. But, we're still kind of getting that paper wrapped up because it's super wild. But, there was something in dollar store.
How much
made the difference?
How much copper copper particles had to be embedded into the mixture to make it work?
I think he used like a 5 millimolar 10 millimolar about of copper. It wasn't, I mean, these are nanowires. So these are like, so they're super small.
So you would use them for maybe 1 nano.
Yeah, for making circuits.
Oh, there it is.
So the growing, so of course this is like done
in. Could you grow circuitry that changed like sort of like solar energy and stuff like that, that you could, it was small enough that it could.
It theoretically, yeah. I mean, my thing is we just make it and characterize it, and then
you discover it and go, so you guys do something with this.
Give it to some engineer. Can you make use of it? Because if you look at how like, so semiconductor people, that's a kind of a bottom up approach. They build every single layer. There's, you know, dozens of different kind of processes where they deposit a few atoms of this, remove it, put a mask on, and they pattern these chips. It's kind of magnificent through this reiterative process of etching, deposition, masking, et cetera, et cetera. And there's like, you know, each step is 12, you know, and you want a chip gets bigger and bigger, so you make thousands of these things over time. And so the process we have is sort of like you're growing these wires.
Yeah, that's cool.
Which would be like, I don't know how it would fit into that whole process. So
if you could maybe have the process set up where the wires will grow into a certain pattern. Yeah, that's probably how you would do it.
That's what you want to do, is have somebody seed it and see. But that's I don't know.
Do you go and work a lot of days and be like, man, we are changing the world? People don't even realize what's happening, like where science.
Sometimes I do. Sometimes I'm like, they better. I'm just hoping my students are moving forward and getting, because I have a lot of students. And there's a lot of different like projects in my group. But yeah, some of the stuff we found is pretty amazing. Some of the stuff is like unexpected. So we have so cicada wings have cones on them, nano cones. So if you go outside, you see a cicada. That clear part has all these cones. So about 10 years ago, we found the cones. Other groups have found them before, and we were also early on realizing these microbes, bacteria, gram-negative bacteria like E. coli and certain yeast, cellular yeast like Cerevisiae or Candida, would rupture on these cells. So when you put the yeast on the cells on, they would pop. And there's a group in Australia that showed this, and they actually took it. We were working with yeasts, and we found that these surfaces actually change the way the cells interact. They change their metabolism, and they change the way they process their environment around them, such that they're much more sensitive to certain drugs. So we think now that we can have developed a process where we can tease the cell so to be more vulnerable to known antifungal drugs, which is really good because we're running out of drugs that are antifungal. And fungi are kind of nasty.
Is that worse than is that kind of like antibacteria? We're kind of running into just some trouble with that too.
Superbugs and stuff.
Yeah. One thing with fungi though is it's closer to animals than plants. Exactly. So it's closer to us. So it's harder to attack it without attacking yourself.
That's exactly right. That's a big challenge, is because since they're eukaryotic, you're exactly right. Yeah. They wind up, you make an antifungal drug and you find it, it kills it in the dish, then you can go through, but as soon as you put it on animals, it's like, it's
hurting the animal.
Yeah.
And they can kind of communicate. Fungi can communicate. Do you think they would be like, oh, this is what's hurting us? Do you think they could adapt to that? Is that like a thing?
That's the thing is that I gave you the Rosie story that, we sensitize it. There's a whole other side. It looks like
everything
DNA
adapts.
repairs. Yeah. Well, these things have like, the other thing is that adaptation is like the amount of DNA repair enzymes, the expression of those enzymes are dropped significantly. So what we're thinking is happening, if these cells are hitting this crisis, they're more sensitive, sure. Some of them like 10%, 20% when they actually bind the surface rupture. The rest of them start replicating kind of quickly and sloppily, but they're gaining, they're more sensitive to drugs, but they're also
finding how to adapt to the drug.
Exactly. They're developing more variation genetically. So they actually probably have more mutations per cell. And all you need for a microbe is 1 out of billions to be resistant. And within 24 hours or 48 hours, you got as many cells. I mean, bacteria, E. coli divides 20 minutes. Wow. Every 20 minutes, you got a division. So if you just do, in 24 hours, you have billions.
It's like network security. It's like you're always having to change the software to avoid the hackers.
Yeah, it's exactly like that.
It's a constant race.
And so this is something we didn't know.
Could you make the software that it rewrites itself enough that
AI will eventually, and then with quantum technology that's coming out,
How do you know so much stuff, dude?
I'm a technology nerd.
That's what I think.
I'm an entrepreneur though, so it's good to know these things. But I'm a technology and science nerd. So
that's cool, because like you said, that's where it's going though. This quantum stuff. I think the next step for a lot of computing, debatable, is going to this quantum computing, where you're using, instead of electrons, you're using light and looking at how the state of different materials, especially single atoms, are actually affected by the so that we can get like the same sort of information, but literally in atoms.
Yes.
And that's
Neil tried to explain quantum computers to me, and it was
which now now the silicon-based chips are still, I mean, they're so they've gotten so small. They're almost quantum. They're what, 7 atoms? Yes. Like the circuits are 7 atoms apart.
Yeah,
which is unimaginably tiny.
Which is really crazy. You start thinking about individual atoms, and that's where there's some people are working on catalysts too. Like, so to catalyze certain reactions, a lot of our bulk catalysts, like I guess the catalytic converter you use platinum or palladium, those are bulk, right? So you have like, as small as you make it, the only thing that's reactive is the surface one. So you can start making single atom catalysts where you're using one atom, you're saving a tremendous amount of money. So you display this on a shell, on a scaffold, where you have a single atom that's going to react or catalyze a reaction, and the rest of it's going to be basically, just a platform.
Yeah, or like the structure.
Yeah. So I'm kind of amazed at that. So that's nano. It's like being able to kind of
scaffolding.
And that's getting very biological. Right?
Yes. Yeah.
Because that's how enzymes are. Enzymes are like these. There's strings of amino acids that fold up into a specific shape. And usually in the shape, there's a pocket which engages in a chemical reaction if it's an enzyme. That's going to facilitate that. So we're sort of making synthetic or artificial catalysts. And that's one of the big drives for nano is mimicking biological systems, looking at how like an enzyme will catalyze a reaction and then mimicking the three-dimensional space that enzyme is, which is a nanoscale space, but eliminating all kind of the fluff.
So eventually we could just grow our own technology, or your technology will be able to morph into different things probably.
Yeah, exactly.
So what will that mean, being able to recreate that enzyme thing? What will that do? Like
So it means a lot of stuff we do industrially, like to make stuff. You want to facilitate or make different chemical reactions, right? You want to have a platform where you don't have to rely on, like fermentation technologies are big, right? So we use live cells for that. If you can somehow capture the ability to ferment without having the living cell, but you're creating the enzymatic, kind of mimicking the enzymatic activity on a larger scale, or pumping it through some sort of material.
Change water into wine.
Yeah, theoretically.
So I saw something online where it was they did some nano research like that, and they essentially made it to where these walls, they painted over them, and then so to stop public urination. So when people would go and pee on the walls, it would make the urine bounce back like up towards the person.
Makes sense.
So I didn't know if that was like a thing you'd heard of. It's like they put it in the red light district, like France and Germany and stuff, because people were just pissing all over everything.
There's some surfaces which are super hydrophobic, which you can imagine urine's like water. So it's going to just bounce off. There's actually
if you can put a specific texture in the paint to make it bounce up.
Yeah. There's a group at Duke with a fellow from Greg Watson and a group at Duke showed that these cicada wings, this is an Australian species, they put water on it and it would bubble off. It'll actually use a self-cleaning surface.
I've seen that.
Like Rain-X?
Yeah, like Rain-X, but like on steroids.
It just rolls
completely off. So it's super hydrophobic. So anything so we measure hydrophobicity by putting drops of water onto a surface and seeing how perfect a sphere it can make. Super hydrophobicity goes in the realm when the angle of incident of the tangent of the actual edge of that sphere as it contacts the surface is greater than 150 degrees. So, things like Teflon, but if you structure it, like you were saying, you can get these things that are almost like perfect circles. And at this point, it seems like it comes from this kind of lowest energy state. When you put water on it, it's going to ball up and literally gets flung.
That could be on like windshields and stuff too. Like space stuff.
Except the issue is it's so, you take your finger.
And then a cicada runs into it and then busts it.
Exactly. It just.
Speaking of steroids, is there nanotech that athletes and stuff could you do steroids on steroids?
I wonder.
Is there nanotech that an athlete could use to, let's say,
like a swimmer
on the cellular structure in their blood or whatever the thing is, but to take in more oxygen. You know what I mean? And then
I wonder if they could do that.
That would give you more energy and more strength to the more oxygen you could take in.
How you would do that? You might have a better way of like transitioning, like the exchange between oxygen, CO2, and I don't know how you do it. But again, it might be a particle or something. I'm always leery, these nanoparticles and nanomedicine, because you've seen a lot of studies. Every one of those particles, when they show the rat, like they inject it, oh, it's going to the tumor. And then 2 days later, oh, it's in the kidneys. And then a week later, it's still in the kidneys. It's like, and then like 3 weeks later, it's still in the kidneys. And what the hell is it in the kidneys for? And they're like, well, it never clears.
Right.
I'm like, I'm not going to take that stuff. I don't want these particles.
Right.
in my kidney and liver. It's like those things I use.
Well, they're using CRISPR to treat a lot of cancer now, right?
What's CRISPR?
It's just a gene editing technology, so you can go in and find the genes you need to replace or recode, and you change it snips that gene out and replaces it. I think there's something.
Is that what Merlin is? I read something about Merlin. Is that kind of a thing that deals with kind of the same thing like that?
I don't know.
I think they've got a better version of CRISPR now.
Well, CRISPR's
constantly evolving.
Yeah. It's like, I think it's 40 years story, right? The initial CRISPR was like, they found these repeated elements in bacteria like 40 years ago. And it was like, oh, this must be, they hypothesized it was a bacterial immune system for viruses and stuff. And so they went through the entire, and then recently, I think 2012, Duda et al. at Caltech showed that you can take this enzyme, which is like 3 different things, put it into one hollow enzyme and be able to then manipulate, it cuts very specific sequences. So the idea is you could target very specific sequences with these enzymes.
You can even change adult DNA. Yeah, which is pretty amazing.
It's totally amazing.
Are they doing any of this stuff, or this is all just
doing it?
They're all over the place.
They're already regenerating eyes, like completely.
People with like pigmentosa, retinosis pigmentosa, which is a signal mutation. You go in there, fix some fraction of the cells that have this damage, and the person won't be. They inherited this, right? They won't be blind.
They re-inject it with like a benign virus, right? So, so it spreads through the cells.
Can I do something so I can stop wearing reading glasses and my eyes could keep getting...
Eventually, yeah. Soon, probably.
I hope before I'm...
Probably 2030.
Yeah, and I think they're going to start making... They'll start getting it. The problem is it's expensive. It's, again, targeting. How do you get... Like the eye is a good one because it's right there. Yeah. And you can use a virus to kind of dope and get huge coverage of what you need. Blood diseases are a big one too. Like if you're born with a cancer, you could fix the stem cells, fix that. So those are relatively easy. But other types of I think muscular dystrophies are another relatively easy one because you could target specific muscles. But it's the challenge is targeting, and that's where a lot of other efforts, whether we talked to nanobots earlier, will theoretically be used to target specific cell types.
So how is this country ahead in nanotechnology research than other countries, or are we?
I think China's really China's just cooking everybody.
Right.
They have so much money and so much stuff. I mean, you're starting to see, they actually are thieving. There's a nanoscientist at Harvard, got kind of, what's his name? I forget his name. He's indicted now because he's has some sort of collusion with China or presumed or.
Yeah, I mean, if I was China, I would be pouring money however I could into every place in America that's got nanotechnology research happening. And
they have their own. I mean, they have so much money research-wise.
Oh yeah.
You look at their papers. Oh my God! How much money they blow.
The 10th largest.
Do they share their science?
Oh, it's published. I mean, they're publishing furiously. I mean, you look at the journals. And they're publishing. I mean, some of the experiments they do cost so much money. It's just amazing that they're.
But they do it, and then they discover something, and then that's easier then to not have to go do all that. Wouldn't they cut out a lot of that expensive work, and then you're like, oh, cool, we could start using this.
That's for sure. I mean, it's the community of science, so they're sharing it. But as far as like they're really, I think, pushing the envelope. I don't know where we stand.
I think we're still ahead of them a little bit.
Yeah.
About or neck and neck, maybe.
Do we need to be?
I think so. Yeah, I think whoever controls the most powerful AI and nanotechnology controls the world.
Yeah.
Doesn't AI?
So I think really what you want to have is us working with them. And that's that diversity thing.
In the community of science, I think it should be borderless, basically, but it's not.
I agree with that too. But like for going off of this, like you think about that, like nuclear technology, like when we're as a country, like, well, you can't have science that was existed since 1950. Like, how do we? I don't know. We can't, right? It's science. Like, they're gonna have the ability to do these things,
and they're gonna make this stuff to make it, to actuate it. Yeah, I don't know.
This technology is way more powerful than nuclear technology too.
Think so?
Way. Yeah.
I think it's a lot more impact. It's a lot more diverse because nuclear, you're looking at like, splitting the atom and finding energy of being able. Here, there's so many different applications and so much possibility. I mean,
what's intelligent technology?
Yeah. And you're being able to apply it to a variety of different problems, I think.
Is it being used more to like a variety of problems, or is it also a lot of the money being put into how do we weaponize it?
Well, there's that too.
I think weaponizing it. Yeah, of course they're going to weaponize it.
Yeah, it's
Iron Man suits.
Yeah, I wish.
Well, one interesting thing about China is, and the U.S. is typically the West is more innovative, typically, and then the Chinese are better at implementing the innovations or replicating them and manufacturing them. And it's unfortunate we can't work more closely with them because we each have our strengths. But what they do is, that's why we're being hacked every few hours by Chinese hackers.
They're hacking the show right now.
And they'll take information for papers and science from labs.
A couple labs that have been had Red Army officers put in as students or postdocs.
Whoa.
Yeah, I told my students last week. I said, "Boy, it'd be scary, but can you imagine if that happened to me? I'd be shocked and appalled, but kind of happy because you're recognized by the Chinese government.
That's right.
Oh my God, I'm that important. I'm like, I'm so shocked.
This guy knows his stuff.
Going home.
I used to get kind of pissed that I'd work with a comic that I know stole jokes, and I'd be like, he didn't steal any of my jokes. What's wrong with my jokes?
I know. But he was there. And is this what you want? It's like, God.
Yeah, that's true.
Then I asked any of them, any of you Red officers? Because, I won't turn you in yet, but, I just want to know. But
just flattered. Yeah,
flattered.
That you thought of me. Wow, that means a lot.
I'm on a list somewhere in China.
I'm sure somewhere in their training in the Red Army, they were like, if somebody asks, say no.
Yeah.
But what sucks though is if you had
somehow they missed that, and you're like, is anybody in here with the Red Army?
Come on.
I am. Oh, shoot!
Gotcha. It's all right now. Go home.
It would be funny if they raised their hand though, right?
Yeah. See guys with guns come in your office I made it.
It's what's really upsetting though is if you had a Nobel Prize level technology that you had discovered with your team and then it got stolen and produced.
Yeah, it would be.
They beat you to it.
And that happens a lot, even without the sinister stuff. But it would be awful if it was used like that.
Could you use nanobots to in the blood to go throughout the whole system and start to recognize where things almost like a diagnosis?
I think so. I mean, that's what they're looking for. There's a whole series of applications, and one of them is to find. Like I said, hunting cancer cells, right? So when you're, they delay the sensitivity. A lot of cancer tests they'll say you're cancer free. But you got to get every single cell out of your cancer cell out of your body. So one thing would be having a probe of some sort, sequester, find, label those cells so you can find out where, a metastatic cancer cell is. And so, I think, like I said, there's a lot of possibility for nano. I mean, even in clothing, right? We had nano silver for a long time, but there's a whole other things you could do. They're talking about the Internet of Everywhere, so, something that I haven't heard of too recently, but it was a big thing for a while. And that the idea is that paint and any object in a room could be effectively a sensor. So they were talking about having, let's say, a nano paint that's a sensor that would actually collect information, tell you where the temperature was in every single point in the room, where there's leaks, where there's toxic this or whatever, and you'd be able to relay that to some sort of computer. And so the notion is you'd have to have a nanomaterial that would recognize the physiological or the environmental condition you're interested in, and then relay it back to people. So it would use ambient energy to kind of power the whole thing.
So eventually everything comes to life. It's like you're sitting on a sofa, and it's like it finds the hard points on your body. It makes those a little softer.
Yeah, exactly. So you won't have to do, and it goes back to artificial intelligence, having something that can interface with these materials that would enable you not to, because we're limited because we tend to focus on specific things and think about. I think AI, which is going to give us, is unbiased. You're going to be able to kind of it'll just look at stuff and sort things, whether they're real or not.
Sounds like consciousness, because
Well,
I believe consciousness is sort of we're all in it at all times.
Could be a fundamental base of reality. Back to the nano thing with AI.
Do you really want to live forever?
Sure. Yeah. There's always things to do, but I think what will happen.
When you're studying down to these level in what the particles of life and all that stuff. Does it make you less like spiritual or believe in a source or a not necessarily? I think God's is a goofy term, like a too small of a term.
I think the more you
or do you think of it as
goofy?
Yeah.
Yeah.
I think the thing is that it's the more you realize how little we actually know about anything,
right?
And how much out there is. I mean, and then you start thinking about the complexity of the problem. Is just it kind of humbles you, right? You think you have an answer, but you really don't. And then you realize how much there is that we really don't know, and there's a lot more out there. It's
humbling. The more you learn, the more humbling it is.
Yeah. I mean, we go in, we find something. And then you're like, for instance, one thing we're studying is nanoplastics. So we're looking at nanoplastic interactions with microbes. And you'd be amazed if, there's no impact on the viability of these cells. And yet there's an entirely different transcriptional system. The genes they express are completely different than if other nanoparticles or no nanoparticles at all. And so there's this whole realm that we just don't get that, when we use so many plastics, they mechanically break down. Almost every biome on the planet has nanoplastic in it that we're looking at. We don't know the impact. And we're looking at things that are the base. So microbes are the basis of microbiomes. We're basically carrying 100 billion cells in our bodies, right? 10 pounds of microbes. We're basically a cruise ship for microbes. Yeah. And we don't know how we're impacting them. And there could be like silent death, right? We could be wiping out entire species of really important microbes that could be actually influencing how life on this planet. Yeah. So there's this so you start thinking about, well, we're not even looking at that, right? We're concerned about other things that are more tangible to us, like,
how do we make this tennis racket lighter?
Yeah,
it's
exactly
right.
I need a new fishing pole. Exactly. That type of thing where it's like, you don't really think about the impacts of these stuff, but it's kind of scary.
And how do you attract the young scientists and the young minds, and how do you attract them, or are you having to try to go get them and bring them to this field, or are they coming to you and you're like, all right, we're going to?
Some come. I mean, nano is hard because it is this convergence of multiple fields, and so when we recruit, the challenge we have is that you don't recruit. We try to recruit from chemistry, the traditional sciences, but we're asking them, try this out. You're in a department now that has physicists, chemists, computer scientists, biologists.
I think that's great.
And it's really great, but a lot of people aren't willing to take that risk. So, and actually in America, there's actually a push where a lot of people, the very smart people, don't want to go to grad school. You can get kick-ass jobs as a startup company, doing really creative work, really creative science and engineering, where you don't need an advanced degree, where you kind of say, okay, I'm going to spend the next 5 to 6 years of my life toiling away to get 6 or 7 papers out, get a dissertation, go through all this kind of formal rigmarole where.
Oh, I just have this image of of scientists just like they're driven by the curiosity of science, and as opposed, I guess we're all the same. Yeah. We want a job, we want a thing.
Now these people are very keen on jobs. I mean, there are some people are. I mean, there's but they're somewhat rarefied, I would say, compared to the normal people want. I hear a lot of how do I have work-life balance? I'm like, well, when I was in grad school, I just
all work.
Work is life.
Time. And I went to the gym 4:30 to 6:30 I'd work out, and then I would go, and then from 6:30 till 8 I would go, and then I'd go home and sleep, and then you go out, you have fun.
Because it's not really work, right? It's not like a job. No, it's what your mind is sparked to do.
Yeah, and you're just completely involved in trying to solve these problems and stuff.
Is it still that way for you, or is it because I'm the same thing with comedy. Like I want to do this, and I can't not do it, and then I'm like, there's a part of it that's like, oh, this is the job, right?
Yeah.
And so as long as I can keep that spark of I love this.
Yeah, I have that spark, but unfortunately, I'm managing a lot of students now, and if I think if I had fewer students, I think I have 20 students now, which is way beyond my capacity.
Right.
But I'm doing it, no stress. It's all good. But yeah, I'd rather have, I really, there's something, I enjoy myself in the lab doing the work individually with my students or by myself. Plus I know I can do it.
I don't know how you do it. Your job is to inspire other people's minds to want to do the science that you can't do because you have to teach them how to do
exactly.
it. You want a student to become you, like that is inspired to want to do this research and this work, right?
Yeah.
Which you're not doing because you're wasting your time on these clods.
And then exactly. Well, but you also they're not you, so they don't have your skill sets, because there's a bit of skill involved. I mean, there's a skill of like how you approach the problems, but technically you have to have pretty good hands. And the better students are the ones who understand kind of fundamentally how things work. So
and then give you a different perspective too.
Yeah, great chatting with you guys.
Yeah, I enjoyed it. Thanks for coming in, Neil.
Yeah, my pleasure. Yeah, absolutely.
Yeah, if you guys ever want me back,
of course I do.
I'd love to come in.
Yeah, that'd be awesome.
That's the idea of this show is me learning stuff, or at least talking to people that do, and so that other people can. Yeah. I got no answers for anybody about anything, so. I'm unlike
I
most
feel
podcasts.
the same ways. I don't have any answers either, but I don't mind talking. Illusion.
Yeah, or if you have some cool things happen, or if you're like, I want to get Tom's 7 viewers to get behind me on my push for funding.
Yeah.
I want to hear your pitch for those things.
That's not good. I can give you some pitches.
Yeah. All right. Well.
Yeah, this is awesome.
Thanks.
Thank you.
Yeah, thanks.
Thank you.
Is that good, Seth? We good?
Yeah, it's groovy.